This video presents a set of 4 experiments used for identifying functional HIV-1 Nef protein motifs and for developing peptide-based inhibitors. Specific short peptides derived from motifs found in full-length proteins both retain their biological function and competitively inhibit the function of the full-length protein. To identify apoptotic motifs in HIV-1 Nef, Jurkat cells are exposed to Nef scanning peptides, and a tunnel apoptotic assay is performed to identify peptides that induce apoptosis. A second set of peptides containing variations on a Nef secretion motif, the secretion modification region, or SMR, are transfected into cells along with Nef GFP. Loss of function due to competitive inhibition, indicated by decreased secretion of Nef GFP, is assessed by fluorimetry. To identify cellular factors that interact with the secretion modification region, or SMR of Nef, co-immunoprecipitations are performed using SMR wild-type Nef as bait. Finally, to test whether these interactions affect secretion, antibodies against identified binding partners are transfected into cells to assess whether they antagonize Nef motif secretion. The results obtained identify 2 functional apoptotic motifs in Nef